Welcome to the debate. Today we're delving into the really intricate genetic identity of modern Mexico. It's a population shaped obviously by thousands of years of deep pre-Columbian history, and then followed by these huge demographic shifts during the colonial period.
Absolutely. And the source material we're looking at paints a clear picture, doesn't it? It's a mosaic. You've got indigenous American roots, European ancestry, mainly Spanish, of course, and also African ancestry. And what's fascinating is how much this varies across different regions of the country.
Exactly. So the central question we're tackling today, the core of our debate, is this. Is the genetic identity of modern Mexico best defined by the overwhelming persistence of its maternal indigenous American heritage? I mean, it's almost universally preserved. Or is it better characterized by the really profound, regionally varied admixture that defines its nuclear genetic structure, the autosomal and the paternal lines?
A key distinction.
Right? And I'll be arguing that the foundational, the sort of unifying identity of Mexico really rests in that stunning persistence of its maternal indigenous American legacy.
And I'll be taking the view that while that maternal line is absolutely crucial, the defining characteristic, the thing that really tells the story of modern Mexico, is the complex, heterogeneous admixture and that strong sex bias that's written all across its functional nuclear genome. That's what dictates the huge regional differences we see today.
Okay, so let me lay out my position. The most, I think, foundational and stable signature of Mexico's modern population is this enduring continuity with its indigenous past, specifically through the maternal line. When researchers look at mitochondrial DNA, mtDNA, which, as you know, is passed only from mother to child, they consistently find that somewhere between 85 and 90% of maternal lineages in the general Mexican admixed population, the group often called mestizo, are of Native American origin. And these lineages, they belong overwhelmingly to those four core Pan-American haplogroups, A2, B2, C1, and D1. Now, these aren't just random letters. They represent the distinct genetic footprint of the founding populations that first settled and expanded across the Americas. This incredibly high frequency of indigenous maternal ancestry, well, it contrasts really sharply with estimates for overall Native American ancestry that you get from the rest of the genome, the autosomal DNA, which averages, somewhere between maybe 30 and 66%.
Quite a gap.
It's a huge gap. And this disparity is basically a clear genetic marker of historical directional mating, meaning immigrant European man predominantly paired with Native American women during that colonial period. And this, I argue, makes the maternal lineage the strongest, the most stable, and the most consistent genetic anchor across the entire country, right from the time of the conquest onwards.
Okay, I understand the strength of that maternal anchor argument. I really do. But defining Mexico's identity solely by mtDNA, well, it kind of ignores the biological and, frankly, the demographic reality that's encoded in the nuclear structure. The nuclear genome, which holds, the vast majority of our functional genes inherited from both parents, it tells a fundamentally different story, and a far more complex one. So, while the maternal line is overwhelmingly indigenous, as you said, the paternal lineage, the one we track through the Y chromosome, passed father to son, that's predominantly European. Studies show contributions ranging anywhere from 48% up to a staggering 89% in various admixed populations. This striking sex bias isn't just a detail, it's central. It's the genetic fingerprint of conquest and colonization.
Okay.
Furthermore, the European contribution across the entire autosomal genome isn't trivial. It's substantial, and it varies dramatically by region. You're looking at roughly, say, 60% on average in the north, maybe 40% in the center, and perhaps 30% down in the south, based on some of the big analyses. This level of varied, multi-continental admixture, including, importantly, a small but historically significant African segment, usually around 2 to 6% of the overall autosomal genome, this is precisely what defines the modern complexity and the demographic history of the Mexican population. So to ignore the nuclear genome, I think, is to miss the defining admixture event entirely.
I see why you emphasize the autosomal and paternal picture. I mean, it certainly reflects the profound demographic shock of the colonial period, no question. But let me offer maybe a different perspective, one that's rooted more in deep time. The persistence of that 85 to 90% Native American mtDNA across such a wide geographic and demographic area, well, it's more than just a data point. It's a sign of unbroken, fundamental continuity with the nation's earliest human history.
Deep continuity.
Yeah, I mean, when we look at the coalescent age estimates for those core Pan-American haplogroups, we're talking about lineages that started expanding really rapidly into the continent right after the last glacial maximum, maybe 20,000, perhaps 16,000 years ago. This maternal legacy survived for millennia. It survived the catastrophic population decline, the bottlenecks associated with European contact. It demonstrates a fundamental genetic resilience. So if we're looking for the defining, the unifying character of the Mexican population, what connects a mestizo family in Sonora to one down in Chiapas, this shared maternal legacy, I argue, is the strongest, most consistent signal. It sort of acts as the shared foundation upon which all the subsequent male lineage replacement and autosomal variation actually happened.
Okay. That's a compelling point about the deep continuity. It is. But I would argue that the massive sudden split between the maternal and the maternal ancestry isn't just like a footnote to history. It is the fundamental mechanism that created the modern Mexican genome we see today. This extreme directional mating, that's the defining event, in my view.
Okay.
The high proportion of European Y chromosome lineages combined with the regional appearance of African maternal lineages, specifically those L hap groups, L1, L2, L3, which you find concentrated in coastal states like Guerrero and Veracruz, well, that highlights the multi-continental dynamic. Historical records confirm these areas were main entry points for African slaves. So the genetic identity is inextricably linked to this three-way admixture: indigenous, European, and African. And it's linked to the highly directional mating patterns that drove it. This complex mixing, the very definition of mestiçage, that's what defines modern identity, not solely the pre-Columbian maternal inheritance. And furthermore, I'm sorry, but I just find it difficult to define the identity of an entire nation based on a singular maternal continuity, especially when you consider the sheer genetic variation within Mexico itself. This internal stratification is paramount, especially when you think about practical applications, like public health, genomic medicine.
Sure. The regional differences matter for that, yes.
They matter immensely. We quantify this variation using tools like principal component analysis, PCA, and FST calculations. PCA, is a statistical method that basically plots genetic relationships, showing how closely groups cluster. FST, on the other hand, measures the proportion of genetic variation found between populations rather than within them. It's like a gauge of genetic distance. And what the data shows is dramatic stratification. Northern states, like Sonora and Zacatecas, they cluster genetically much closer to European reference populations. Their mestizo populations show higher heterozygosity, meaning greater overall genetic diversity. Meanwhile, southern regions like Guerrero and Yucatan, they cluster closer to Amerindian groups. The FST value measuring the genetic distance between, say, the Zapotecas in the south and the mestizo populations of Sonora up north is notably high. I think one study reported it around 8.2. Now, to put that distance in perspective, that represents a really profound genetic divergence between populations living within the same modern country. This north-south gradient, reflecting unique local indigenous histories and different intensities of colonial settlement, that is the decisive feature for understanding Mexico's complex identity today.
Okay, that's a compelling argument, and I absolutely agree. The importance of regional stratification for applied genomics for public health, it can't be overstated. But have you considered that this dramatic regional substructure, it occurs within the context of that overwhelming indigenous maternal foundation? While the paternal and autosomal lines, yes, they vary dramatically from north to south, creating the stratification you just described. The underlying maternal heritage is consistently overwhelmingly Native American, right across the board. Within the context.
Yes. When we analyze molecular variants using AMOVA, that's analysis of molecular variants, on the mtDNA across those macro regions you mentioned, north, center, south, the results are pretty powerful. The vast majority of the observed genetic variation, we're talking over 94%, is accounted for within these regional subsets rather than among them. Now, that statistical result strongly suggests that beneath those significant autosomal differences, there is a fundamental shared maternal basis that actually unifies these diverse regions.
Okay.
And furthermore, if we think about modern identity in the context of the wider region, comparing the general Mexican population within Mexico to, say, Mexican Americans in the U.S., it reveals the strength of this continuity. Mexican Americans tend to show slightly lower indigenous mtDNA proportions, maybe closer to 82 or 83%, compared to the population remaining in Mexico, which holds on to that higher 87 to 95% native mtDNA signature. This slight divergence, well, it just underscores the strength of that high maternal indigenous continuity as the crucial national characteristic in Mexico itself. It acts as the consistent baseline against which all subsequent and ongoing admixture variations are measured. So, in summary, while we fully acknowledge the intricacies of the sex-biased admixture, and yes, the profound regional stratification revealed by the nuclear markers, I maintain that the enduring and the unifying genetic identity of Mexico is truly rooted in the overwhelming continuity of its indigenous American maternal lineages. This shared deep-time genetic feature speaks directly to the unique demographic narrative of the country. It provides the most prominent and consistent signal connecting the present population structure all the way back to its profound pre-Columbian past.
And my position remains that the true complexity and therefore the defining feature of the Mexican genetic identity really lies in those intricate, regionally variable admixture patterns revealed by the functional nuclear and paternal markers. The sex bias that established European paternal dominance and that profound north-south stratification, which is visible through differential diversity measures like PCA and FST, these aren't secondary points. They are essential mechanisms for understanding modern demographic history, for addressing population health disparities, and, crucially, for developing effective genomic medicine strategies for what is a deeply diverse and historically layered population. The material, I think, clearly illuminates the value of considering the total three-dimensional complexity of the nuclear genome to fully appreciate the dynamic, ongoing narrative of the modern Mexican population.